Hello, everybody. It's a pleasure to be here today to be on this very important virtual meeting for metastatic breast cancer. I would like to thank, first and foremost, Sharon Schlesinger for inviting me to give this talk today. She is a dear friend and a kindred spirit, and we've been working together for the last couple of years on an important project, which I will touch on at the very end. But what I really want to talk to you about today is why it takes so long to see progress. So from bench to bedside, why does it take that long, as long as it takes? And really, we can sum it up in this image right here. I almost don't have to say anything else. So it's a very, very complex process with a very, very poor success rate. And I'll explain a little bit why that is in this talk. So first, I want to start by telling you how long it actually takes. It takes an average of 10 years or more, some estimates up to 12 years and even longer, for a new medicine to complete the journey from initial discovery to the marketplace, with clinical trials alone taking 6 to 7 years on average. And the average cost today is estimated to be $2.6 billion to take a drug target from initial discovery into market. And that's gone up significantly over the last even just 10 years since I've been talking about this. So why? There are many reasons why, and I'll share with you. A very simple and maybe obvious reason why is that cancer disease is complex. We basically have, if we start with a normal cell, we have to figure out how it becomes a cancer cell. And the way that happens, it happens through various mechanisms, a multitude of mechanisms. And then after we figure out why that happens, we have to figure out how to eradicate it. And that's not an easy feat. So just to give you a little bit of an overview of the complexity of human genetic variation that underlies disease is the following. We have so many things that can go wrong. We can have an incorrect number of genes. So if you start off with 2 copies, it's possible for you to lose a copy or both copies through just structural deletion, or you may get more copies of that gene. You might end up with 100 copies of that gene. You might have that gene improperly regulated, so it's expressed and more of that gene is being produced when it shouldn't be produced. It causes tumor cells to grow, for example. It could be through a mutation that also leads to abnormal function. And mutations arise from hereditary reasons, from ionizing radiation, exposure to toxins that we are generally exposed to on a daily basis, structural changes, something called epigenetics, which doesn't involve the structure of DNA, but involves the chemical modifications that allow DNA to be packed and regulated in a certain way, and also abnormal protein function. And then on top of all of that, we have to understand how all of those things are interrelated and figure out what are the genes, the set of genes that cause disease, and which ones are the ones that are the most critical that we can target and inhibit. So this is an overview of the drug discovery and development process. It is really a long and risky road. As I said before, in the United States, it takes an average of 10 to 12 years for an experimental drug to travel from the laboratory to your medicine cabinet. Only 5 in 5,000 drugs ever enter preclinical testing, and only one of those will actually get FDA approved. And so you can see here that the drug discovery phase, people are looking through and trying to filter through thousands of compounds. A few of those will enter into preclinical testing to determine their validity, and then only 5 will make it into clinical trials. And there are several phases of clinical trials, as you can see, 1, 2, and 3. And then you have to enter into the FDA phase. And only one drug from all of that will make it. So that is a very grim perspective. In the preclinical testing phase, a pharmaceutical company conducts certain studies before the future drug is ever given to a human being. And this is a critical step in the process. And this involves laboratory and animal studies that must be done to demonstrate the biological activity of the drug against the targeted disease. So the FDA process is also takes a very long time. Once you have a drug that you want to approve, you have to file for its application. You have to file an NDA, and you have to basically ask permission for the FDA to start investigating the validity and the safety of that drug in humans. And then you have to go through at least, 3 phases of human testing. In the first phase of human testing, you're really typically just testing the safety of the drug in healthy individuals. And typically those studies are small, about 20 to 80 individuals. If that's a successful trial, it advances to phase 2, which begins to test the efficacy of that drug in patients and also continue to assess its safety. Safety is constantly being assessed. And the goal here is to obtain preliminary data on whether the drug works or not. This is really the focus of a phase 2. If that happens, and oftentimes too, it's through a placebo control study, so that there is a control arm not receiving the drug, or at least just receiving standard of care without the drug. In a phase 3 trial, that opens up to thousands of people, and typically the studies are gathering more information about the safety and effectiveness of the study in different populations and at different dosages and different combinations. And once we pass through all of those phases, then the approval process can begin. So the FDA, in addition to now confirming the safety of the drug in humans, has to go through another series of regulatory process that involves reviewing with the investigators everything to date. You have to file a new drug application, an NDA, which is being reviewed. Once the application is being reviewed, the next step is that the drug labeling, and there are a lot of rules around how to accurately and properly and ethically label drugs. Then it's followed by a facility inspection to make sure that everything is done according to the best standards for producing the drug safely and effectively. And then there's phase 4 clinical trials, which is in the post-marketing phase. And this is really established to introduce a risk assessment system and continue to, again, also assess the safety of the drug. But let's get a little bit deeper into the drug discovery process. So I got a lot of these really nice pictures from the website, but specifically this website called Dr. Target. If you're interested, you can go and read about it a little bit more. But I want to take you through, again, the nitty-gritty details of the reasons why it takes so long. So we saw already kind of a brief overview of why, but the very, very first step. Oh, and I should add here that here, as you see all the stages advancing from one to the next, each stage's output really becomes the input for the next one. The system works like a pipeline, each phase feeding the following one with backups and preventions and failures. And the concepts and the theory of the project is also constantly being refined. So here, the process starts with target identification and validation and target selection. So you need to start with, if you're going to get to a drug, the drug is hitting a protein. We have to identify what that protein is. That's the first step. So somebody has an idea of what protein that's abnormal in cancer needs to be targeted pharmacologically. Okay. That process can also be very time consuming. It happens a lot in the university labs, and also pharma is actively looking for new leads as well and targets. But this process can cost millions of dollars and also take years. And the difference between so generally the target is naturally existing cellular or molecular structure involved in the pathology of the interested drug that is being developed. But there are established targets. And what that means is that there's already a lot of information about that gene in the literature. And somebody is out there now trying to generate a drug against it. Okay. And so then there's new targets. Maybe there's a target that has never been described before in disease, and now somebody's going to try to figure it out. So what this entails is it has to be disease relevant. So there needs to be a focus. So for example, let's say we have a new candidate gene in breast cancer. And what we're going to have to try to do is go into the lab and basically manipulate that gene to really understand how it functions, and if it really is involved in breast cancer formation, progression, metastasis. And where that target comes from, I'll explain in a minute, but that target oftentimes comes from genes that we've identified nowadays through genomic technologies. All right, so we're going from and analyzing hundreds and thousands of samples where we say, oh my God, 30% of these samples have an aberration in this gene. And it seems to be correlated to something about the disease, let's say survival, let's say metastasis. And I think it's worth studying. So I'm going to generate a series of experiments that are going to prove my hypothesis that this gene is important in cancer. And this is the beginning of the process. It starts there. And from there, we're going to publish papers, a lot of papers. It's going to take years to do it. And other labs are going to start catching on, perhaps. Maybe other labs are already doing it. So building the knowledge base about the target can take years before anybody is going to be willing to invest billions in developing a drug, because that's even longer and more expensive. So we need to be able to have a process to do that. And the process is just very rigorous experimental research that gets published, and the field decides whether, and pharma decide whether this is worthy of a potential target. It has to be rigorous because, as I said, 1 in every 5,000 to 10,000 targets only make it to clinic. So from the target to a lead molecule. So now we've established the target. We need to figure out how to target that. What is the molecule, the drug that we will use? And those drugs are not always available. So we sometimes have to start from, oftentimes all these drugs, you start from scratch. So the process of finding a new drug against a chosen target for a particular disease usually involves high throughput screening, where large libraries of chemicals are tested for their ability to modify the target. And that process, and I won't take you through every single one of these steps, but it's a long process, an expensive process. And remember, you have to figure out, is the target, now that you have a target, can you find a good drug to inhibit the target that's going to have as few side effects as possible? That's going to be determined by what are other targets this drug might hit. So a drug needs to be specific as possible to the gene that you want to inhibit. It shouldn't be inhibiting other genes. And if it does so, it has to be at a very, very minimal level. But that's what makes a drug dirty, and what increases the toxicity is that if it hits more than its initial target. So there's also a therapeutic window. So you will accept some side effects because the benefits outweigh the side effects, but you still have to be rigorous in that process to make sure that it is as safe as possible for use in humans. Then you have to figure out how to scale this drug, how to mass produce it. And the process, as you can see from here, is you can see why it starts to take 10 years. Drugs in development typically fail in the clinic due to a lack of efficacy and/or toxicity, which are often attributed to inadequate preclinical target validation, and just, it just so happens to be the case. The molecule that people settled on ended up not being the one that would have been the best choice, or maybe there wasn't a better one. So sometimes the drug itself, to improve the efficacy and the specificity, gets chemically altered to the structure gets altered basically synthetically to try to enhance its activity and reduce its toxicity. And so again, it's just all of this should be adding to why this takes so long. Now, drug development is a little bit different than biomarker development. Biomarker development is also something that takes many years, and it starts with designing a study and identifying samples that you can use to identify a biomarker, let's say for recurrence. Then you have to prepare those samples and analyze them in the lab. And you have to process the data. But the sample preparation analysis, sometimes those methods have to be worked out to make sure that the data you generate from that process and from that analysis is robust, accurate, reproducible. And then you have to process that data. You have to statistically analyze it to see if this biomarker is sensitive and specific, so has a low false positive rate and it has a high true positive rate. Then you have to translate that biomarker into clinical practice, for which then you need to validate it in like many, many, many more patients, sometimes hundreds and thousands of patients. And then you have to interpret the outcomes of the data that you generate. And then you have to go back and try to figure out if there's anything that you left behind that needs to be figured out. Okay, so how can we make this go faster? Since the end of the Human Genome Project in 2003, genomics has become the mainstay of biomedical research. And you can see here that this is a timeline of our understanding of disease. And by the end of 2003, you can see that we started to really understand, or we got a good understanding of the structure of the genome and the biology of genomes. And as time went on, the understanding of the biology and the structure of genome led to the understanding of disease, and that advanced to science and medicine with precision medicine. And then with ultimately now 2020 and beyond, the goal is to improve healthcare through genomics understandings. And we're here. We hear about things like personalized medicine and genomics-enabled medicine, and it has been instrumental. And this will help us and is helping us maybe speed up the process a little bit. And it has. And I'll give you an example of how it has. Let's just take, for example, the discovery of the estrogen receptor. The discovery of the estrogen receptor was made in the 1950s by Elwood Jensen at the University of Chicago, all right? And actually, tamoxifen, which was approved by the FDA in 1978, so there's already a 20-year gap. And before 1958, the discovery of estrogen as a steroid of importance was made in the 1920s. It was isolated. People were studying female reproduction, of course, and trying to understand various aspects of human reproduction from ancient times. So we were going from trying to understand a genotype through a phenotype that was being observed. But so in 1920s, estrogen was isolated, and then in 1958, the estrogen receptor was identified. And then it was in 1978, tamoxifen, which was the first approved drug for breast cancer treatment as an anti-estrogen, was approved. And that was actually even accidental and not even intended, because tamoxifen was intended to be used as a fertility treatment that ended up not working well. And if it wasn't for Walpole, who was a reproductive endocrinologist who led this research on tamoxifen, insisting that he needs to study it for breast cancer, because they didn't want to, because nobody cared about breast cancer therapy in the 1970s. And it was because of his persistence and threatening to leave the institution that it ultimately was studied as a breast cancer treatment and then later approved. So it took at least 20 years for that to happen. So at least we're getting, we're now at 10 years. We've shortened the time. And that is because of translational research and the genomic revolution, which allows us to get to the clinic a little bit faster, because instead of going backwards from the lab to the clinic, we're starting from the clinic, meaning we're studying patient samples and we're studying um, uh, making discoveries directly from patient samples and understanding and determining relevance much more quickly than we used to. And we have access to technology that allows us to study, thousands of genes at a time as opposed to one at a time. So this has helped to speed the process up. It has not made it cheaper, but just helped to speed the process up. And so translational research really exploded with the advent of genomics, and which is the process of taking a laboratory discovery to the bedside, to the patient bedside. But actually, it's really more than that, because it's bench to bedside and back to bench. Because once we make discoveries and take them into the clinic, we go back to the bench to study that even further, to continue to refine. And ultimately, and I'm going to end here really quickly because I think I'm running out of time. Ultimately, this is made possible because of the generosity of individuals with cancer who agree to donate their specimens for research purposes. And I would have to end by telling you about what I've done with Sharon, actually, to try to give that information back to the patients as to what we're doing. We're trying to create a system, and we have created a system that helps the benefactors of research understand why it's taking so long by increasing the transparency. And so I launched an initiative a couple of years ago called the Bench with Bedside Initiative, and it's all about empowering with knowledge, hope, and increasing transparency of research. And I'm going to basically stop here, but just to quickly tell you that the goal of this is to help you understand why research takes so long by bringing you into the process and teaching you about the process and keeping you informed of the process to the best of our ability. And with that, I want to end because I think we don't have any more time. And if there are any questions, I'd be happy to answer them. I hope you kind of understand a little bit why it takes so long now.